לא צריך תיקון.
אבל גם עשיתי להיכנס מאוד מהר גם עם קיי לוי.
כן? מה סביר כזה?
איזה 3-4 התאהבתי את זה ממש כאילו. עונה.
זה תלוי בזמן, אבל אני מתקראת משהו.
תלוי בהתחלה.
כן, זה אומר שאת רוצה בוא נתקראת?
נראה לי שכן.
ביחד.
כן.
בסדר.
יותר קרה מהמדורה.
כן, סופר קר. בחוץ גם, אני לא יודעת איך ללכת בלי זה.
תפתחי לו מחר.
אמרתי לו אוקיי, אז נעשה את האיזו שעה שתתת לו היום. לא תשובה בסדר.
אז תתחילי לעבוד על זה.
טוב, מזל שהקדמתי. מה זה? כן, כאילו, יש לי שני דברים שאני עושה בגבעת רם. אחד זה פסיכוביולוגיה ואחד זה בעצם. כאילו, אני יודע.
סתם כדי לאנשים עבור איזה שם. זה נראה כמו שלי. זה מתכנס ממש מהר. זה לא. הייתי בסדר. אני לא בכלל שלי בסדר. כאילו, מתכנס לי נורא מהר גם לאנשים אחרים. כאילו, לא מגיע למטרה שלו.
כבר תסתדר עם הלחץ של זה. לדעת שזה תתקצר.
לא, עדיין תפס לי לא תיכנס. אמרתי, זה 50 זה מתכנס כאלה 3. לא יש לי עוד פעם שזה ראיתי שלא צריך שזה מתכנס מהר מדי כדי שאני אסתכל את זה.
מה זה? אתה מתחבר בשלך? לא. אני לא... יש סמק החדש? כאילו... לא, לקנות כבל. ימי או משהו. כן. לא, פה יש לך... Okay, so just Yoni is from the medical school and it's the neurobiological for medical neurobiology.
Medical neurobiology department and he's going to talk about reward, right?
Addiction.
Addiction and reward. Alright.
Okay. Do I need to use this? Yes? Okay. Just a second. Good. Not okay. My name is Yoni Kupchik. I'm from the medical school, from the Faculty of Medicine. My lab does, we try to understand the reward system, motivation, and especially what happens in the brain in extreme cases of motivation, like addiction. Today I will talk about drug addiction and kind of a story that I've been involved in during the last 4 or 5 years. But just to show you what we do, so we have in the lab 3 main research pathways. One is we try to understand the physiology of the reward system. We specifically are interested in the pallidum, in the ventral part of the pallidum, and trying to understand how it is involved in the generation of motivated behavior. So we're doing basic stuff to understand physiology. We also have two models of extreme motivation. One is drug addiction, mainly cocaine, and the other one is obesity, which I will not talk so much about today, but we are trying to understand whether obesity could be a result of a pathology in the reward system, which is controversial. Not everyone agrees with it. These are the tools that we use. So we're mainly patch clampers. So we do mainly slice electrophysiology with all the optogenetics and looking at specific cell types, specific inputs. We have a lot of behavioral models, circuitry mapping, all the usual that people do today. In vivo part that we're starting now. So this is what we do. So that was just to let you know what we're doing over there, over the מעבר להרי החושך in Ein Karem. Let's see if at least this works. נכון. קצת So drug addiction, did you hear already about drug addiction or stuff like that in this course? Or one paper of Flusher. Okay. So yeah, I will not talk today about anything that Flusher did, but yeah, he's mostly interested in other regions. But drug addiction, you all know it's a big problem of the modern world. About 10% of people in the world, of adults, use some kind of illegal drug. Mostly marijuana, but also other types of drugs, mushrooms, cocaine, heroin, stuff like that. And the main problem of drug addiction is its relapsing nature. Okay, so many people, not all, but some people that use the drug for the first time will continue using it, and eventually will develop a dependence on the drug. And the main problem of these people is that even if they try to quit the drug, most of them will fall back to use the drug. So something is changed in the brain that makes them still want the drug even though they were off the drug for a long period of time. And when you look at drug addiction from this perspective, so it's like any other relapsing disease, like hypertension, asthma, diabetes. So also drug addiction, 40 to 60% of people that quit drugs go back to drugs within 1 year. This is an average. So for some drugs like alcohol or heroin, about 90% of the people fail to quit the drugs. Now, drug addiction can be looked at as a three-stage process. So first of all, of course, you have the first encounter with the drugs. The first 1, 2, 3 uses of the drug. This is called stage 1, acute drug effect. And you can see that drug use, even the first one, causes some changes in gene expression in the brain, but those changes will disappear or will go back to normal if the person does not use the drug one more time. Okay? If a person starts using the drug on a regular basis, then it goes into stage 2, the transition to addiction. The transition to addiction, you see an increase in the changes in gene expression that go together with increased use of the drug. But this is not addiction. This is not the clinical addiction yet. The end stage addiction appears only after withdrawal is started. Okay, so the final change in the brain that stays forever is induced by the withdrawal. Okay, so in a sense, if a person uses drugs and doesn't quit or doesn't try to quit, the brain is not addicted yet. So what causes the addiction is the try or the attempt to quit, and then the brain has to adjust to the lack of dopamine or
Addiction fully defined by this gene expression?
If addiction, you mean clinically?
Yes, because
No.
You define addiction according to gene expression.
No, so this is not a clinical definition. It's just a way to show how addiction develops. No, addiction. If a person goes to a clinician, psychiatrist, so he's already here. The addict, and then there is the DSM, the manual of psychiatrists. So there are 11 criteria that if you have 2 of them, you can be diagnosed as an addict or dependent on a substance. Many times you have more than that. You have 4, 5, 6, 7 criteria. But this is just to show you, in general, what happens in the brain. So it's not a clinical.
Can I ask, if we talk about the brain,
Yes.
the gene expression fully defines the addiction?
If
Like,
it
is it a one-to-one relation between the
well, everything that I will show you, for example, today, is changes in synaptic transmission, which is driven by gene expression. More AMPA, less AMPA receptors. So it depends on what gene. I guess some genes can be correlated better than others to the development of addiction. Yes.
More general, most of the research that
Yes.
But it's not the most addictive substance, or like,
So you ask
why
why
opiates or like,
yeah, why do people, most people do cocaine addiction, including us?
yeah,
That's your
but
question.
it's not maybe I'm not up to date with the numbers.
No, no, you're
It's not the largest addiction.
yes. There are two main reasons for that. One is political. So in the 80s, that's when the NIH, the National Institute of Health in the United States, started to fund addiction research in big money. And the main problem in the 80s was cocaine. Well, the main problem of the rich people was cocaine. And so most money went to cocaine research. This is a little bit about the politics of science. And the second reason is that it's easy to make an animal, it's easier to make a rat or a mouse addicted or dependent on cocaine than on heroin, for example. And also marijuana, but that's maybe reasonable, I mean, understandable, but it's easier. Cocaine is more immediate, and you don't have, with heroin you have a lot of you have to start with a high dose, then you have to go down to a low dose, then you have to, it's more complicated. Now people do also heroin, but once the big group at the beginning did cocaine, then everyone compares themselves to cocaine. But I will show you today really a little bit about other drugs. But mostly people do cocaine for those reasons. Now, one of the things that people see is that the role of dopamine is important mostly in the beginning of becoming an addict. So when you use the drug the first time, second time, third time, you have a rush of dopamine that gives you a good feeling, hedonic feeling, and this is the cause or what drives the person to use the drug again. But the influence of dopamine gets lower when you get more and more addicted. So, for example, in addicted animals, you don't see a lot of dopamine released in the nucleus accumbens after they went through withdrawal. So they're not enjoying it anymore. It doesn't give them a good feeling. It's just they need it. They need it because dopamine drives changes in gene expression and other changes that eventually affects other systems, like the glutamate system or the GABA system, and these systems encode or engrave the long-term changes in the brain of an addict. So I don't know if most, but a big part of addiction research is actually on glutamate and GABA neurotransmission and less on dopamine, but there is also a lot of on dopamine. So what we're interested in is trying to understand what are the changes in the brain, in the nucleus accumbens, and later the ventral pallidum that cause the addict to be vulnerable to relapse. What is different between the brain or the reward system of an addict that will cause this addict to fall back to drugs compared to normal animals? And the main perception in the field is that the drugs hijack the brain. Okay, so we're trying to understand what does it mean to hijack the brain, and by that, maybe in the future, we will be able to help the brain change and fight addiction. So this is the basic, very simplified circuitry of the reward system. We're talking about mainly three structures: the nucleus accumbens, which is the input structure of the reward system, or the basal ganglia, the ventral part of the basal ganglia. And from the nucleus accumbens, the information flows to the ventral mesencephalon, the VTA, the substantia nigra, and it flows to the ventral mesencephalon in two parallel pathways. One is a direct pathway. It's GABA. Yeah, it's all this is GABA. So GABA projections to the ventral mesencephalon. This is the direct pathway. Activation of the direct pathway is thought to drive addictive behavior or motivated behavior. And there's an indirect pathway that goes first to the ventral pallidum, then the ventral pallidum goes to the ventral mesencephalon, and activation of this pathway is thought to modulate or inhibit motivated behavior. And what determines if the system will be activated or not is the glutamatergic input that comes from many places in the brain, many cortical and subcortical areas like the medial prefrontal cortex, the hippocampus, the amygdala, the thalamus, and more. Now, you probably know all this. I'll go through it quickly. You know this, right? The direct, indirect, D1, D2. So you probably know it more in the context of motor behavior. But the main thinking until recently was that it is the same also for motivation, because it's striatum and pallidum, so it's probably the same as the dorsal striatum and globus pallidus. So the reason that the direct pathway causes behavior is because it disinhibits the thalamus. Right? So the thalamus generates behavior, and it is under tonic inhibition from the ventral mesencephalon. So when you inhibit the ventral mesencephalon, you actually disinhibit the thalamus and behavior. And you have, in the indirect pathway, you have GABA, another GABA synapse that flips the signal. So you have three inhibitions. So overall, it inhibits the thalamus and prevents or inhibits motivated behavior. Now, the important thing I wanted to show here is that it was thought until recently, again, that the direct pathway originates in medium spiny neurons in the nucleus accumbens, expressing the D1 dopamine receptor, and the indirect pathway is from D2 medium spiny neurons, the medium spiny neurons that express the D2 dopamine receptor. Okay, so there is a differentiation between these two. Okay, so why is relapse so easy? Many people try to understand that, and even I don't know if you remember, you know this actor? I don't know if it's too far back. Yeah, so he relapsed to heroin. I think it was after 20 years of being clean, and then overdosed. So why is it so easy for them? And the way we try to understand it is by an animal model, the relapse or the cocaine self-administration and extinction model. Pretty simple. You take an animal, a rat or a mouse, you implant it with a cannula, so the animal can press a lever and self-administer or self-infuse cocaine or whatever you put here in the syringe. The animal has an active lever that delivers the drug and associated sound and light, and an inactive lever and you can see that the animals pretty fast learn to press the active lever and they don't touch the inactive lever. Then you can do many things. So you can you want them to have a withdrawal period. So to make them stop using the drug. So one way is abstinence. So you just leave them in the cage and don't let them get into the box. The other way is give them extinction learning. So you put them in the box, but this time they don't get the drug anymore. So when you do that, you see that on the first day you have a burst. They want the drug, but they don't get it, so they press more. But eventually they will stop. They will stop pressing the lever. And at this point, this is the point that we are interested in, because the question is, did they stop pressing because they don't want the drug anymore? They're healthy again. They're not dependent. Or maybe they stop just because they don't know how to get the drug. They know that this lever will not give them anything, but the urge to get the drug is still there. So if we, at this point, we can put them back in the cage and give them the cues that were associated with cocaine here. Okay, so we don't give them the drug, just the cues. If we do that, they will see the cue, they will think the drug is available, and they will relapse. Okay, and I'll show you now what happens in the brain at this point and after or during relapse. Yeah.
During the reading state.
Yes.
So you use the cocaine or not?
No.
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 Just the cue.
Just the cue. Yes. So we use most of what I will show you is from slices. We compare synapses. First, I will show you what happens in the nucleus accumbens. So the nucleus accumbens gets glutamatergic input. We patch cells in the accumbens and measure the AMPA to NMDA ratio. Yes. Which part? We do the core. AMPA to NMDA ratio, so you can record when glutamate is released. You can record the AMPA current, and you can record the NMDA current. And the ratio between them tells you that if the ratio changes, so the synapse is changed, if the ratio gets bigger or higher, it is considered as a stronger synapse, although, I mean, that's what people like to think about the AMPA to NMDA ratio, but because they claim that NMDA doesn't change. Yeah, it's there only to induce learning, right? To recruit more AMPA. So if there's a change, probably AMPA changed. We know it's not true, but people, I mean, that's only the interpretation. If there is a change in the ratio, so the synapses change. So first thing we did was to check if really there is a change in the nucleus accumbens, in the glutamatergic input to the nucleus accumbens after extinction. So we did self-administration and extinction of cocaine self-administration. And you can see that the AMPA to NMDA ratio is bigger. So this is a saline animal that didn't get any cocaine. This is extinction. After extinction of cocaine, you see that the ratio is bigger. From other when we checked we and others, when you check the level of the AMPA of the protein itself, you can see that it is really an increase in the AMPA receptors in the synapse. Okay, so you see more AMPA, maybe also less NMDA or less well, I'm not going to go into it, but the type of receptor is also important for the current. Anyway, the synapse is changed permanently. Yes, the animals here, they didn't have cocaine for 2 weeks. So it's not a direct effect of cocaine when we record. It's something that cocaine did to the brain 2 weeks earlier. Hmm? Permanent, the longest time that I know of that people check this is 3 months. No, not longer, but it didn't change. It stayed pretty stable.
These are mice that were like in a cage with no other stimulation.
These are rats. Yes, they were a single house, so they didn't have.
There was no other interesting input going on. Like, do people check in like more interesting environment to see? Like if you put toys or.
Yeah. People, not in the context of synaptic, of AMPA and NMDA, people check whether housing animals in single, without friends, causes, makes them more vulnerable to use the drug. But not at the synaptic level, not that I know. I think it does have an effect because putting rodents like mice or rats alone in the cage is stressful for them. Yeah, it has to be taken into consideration. Another thing that happens to the nucleus accumbens is not only plasticity, but also there is metaplasticity. I don't know if we talked about metaplasticity, but so plasticity is changes that happen in the synapse, right? The synapse gets stronger, weaker, more AMPA, less AMPA. Metaplasticity is the change in the threshold to have plasticity. So in a sense, it is the plasticity of plasticity, or how easy it is for the synapse to change. When you look at the synapse, so obviously if the synapse is stronger, if there is more AMPA, you would want to cure the problem by weakening the synapse or inducing long-term depression. But when people try to do that, they found that while you can easily depress these glutamatergic synapses in the nucleus accumbens in saline animals, in normal animals, it's much harder to do that in cocaine animals, extinguished cocaine after extinction. So this was done in slices. Here it's not self-administration; it's just injections of cocaine into the IP to the mouse. And this is an in vivo experiment. So they gave the protocols for both long-term depression and long-term potentiation, LTP. You can see that cocaine animals show a much weaker ability, much lower ability to change, to change the glutamatergic synapses in the nucleus accumbens. So this means that not only that the glutamatergic input, the excitatory input to the nucleus accumbens is strong, is too strong or pathologically strong, the synapse is also stuck. It cannot be changed. And it is thought that the strong glutamatergic input is underlies the increased motivation to use the drug in addicts, and the stuck synapses or the metaplasticity maybe underlies the inability of addicts to change their behavior. So this is the state of the reward system or the nucleus accumbens after extinction. Now, what happens during relapse? If you take the same rat, yeah, well, not the same, but same protocol. You take the rats and give them a reinstatement session, and what we did here is to give them 15 minutes. Okay, they entered the cage and started pressing the lever. 15 minutes, then we took them out and prepared slices, assuming, of course, that the brain doesn't change after. I mean, we tried to do this as fast as we could. We prepared slices of the brain of these animals, and when we did that, we saw that the AMPA to NMDA ratio further increases after or during reinstatement of cocaine seeking. This increase, I didn't put it here, but this increase, the reinstatement increase, was transient. So if you let them reinstate, you put them in the box, but instead of 15 minutes, you give them 2 hours, and then check the brain, you will get AMPA to NMDA ratio that is back to the new baseline, the extinction baseline. So there is, it seems like there is a long-term, consistent effect, increase of AMPA to NMDA ratio after withdrawal, and there is also, during relapse, there is an increase, a transient increase in the AMPA to NMDA ratio, and when we correlated the AMPA to NMDA ratio of each rat to the number of lever presses during these 15 minutes, you can see that there's a positive correlation. So the strength of the synapse, or the magnitude of change in the synapse, correlates with the actual behavior, with the number of lever presses. Now, another group in 2007 did something similar. This time it's not self-administration, it's just 5 injections of cocaine, then 2 weeks of withdrawal or abstinence, and then they recorded either before or after a challenge. Right? In these experiments, they inject them cocaine to remind them, and this injection induces, well, it is a model of relapse. They don't look for the drug, but they start moving faster. It changes the locomotion. Now, yes.
I think that in humans there's motivation to stop doing this.
Mm-hmm.
Is there any indication that brain is going to stop doing this?
Well, rats, using these simple models, rats have no motivation to stop. Why would they? I mean, it's not affecting their lives. If it does, only to the positive, in a positive way. So no, these models, they don't have this component, which is very important in humans, that humans usually want to stop. There are other models which are now starting to be used more frequently, in which you make the rat or mouse, or you try to make them not want the drug. You can give them a shock, like every time they press the lever, they get a shock, and then cocaine. So they have to pay a price. What?
And that's changing
Well, far as I know, in another type of experiment, not with the shock, but you can get, you have a spread of rats that do press and rats that stop pressing. Those that press the levers, they have stronger AMPA to NMDA ratio, higher AMPA to NMDA ratio. So it looks like it does correlate with the compulsivity of drug addiction. Do we stop at 9:45 and then 15 minutes? Okay. Anyway, so if you look here, this is another model of relapse. Yes?
What you said about higher amplitude NMDA, do you think it's like innate?
It's what? Innate?
Innate, like tendency?
It's hard to say, mainly because you don't know. Maybe with in vivo patch clamp, if someone will do it in the future in the nucleus accumbens, then you can measure before and then after and see. There is a claim that it is innate. We didn't see any. So in saline animals, which is a before group, if there was something there, you should see like a spread or something. Like the variance should be bigger than after. But we didn't see anything like that, so it could be a result of using the drug, but we don't know that. Anyway, the last thing before the break, you see here another model of relapse, but here you can see how after they gave them the challenge, so this is let's say this is a saline animal. This is after withdrawal. Again, you see an increase in AMPA to NMDA ratio. And here they gave them part of the mice got a saline injection. It didn't really affect the AMPA to NMDA ratio. But when they got the injection, so when they relapsed, they actually saw a decrease in AMPA to NMDA ratio. So why do you think here you see a decrease in AMPA to NMDA ratio in relapse, and here you see an increase in AMPA to NMDA ratio? These are two relapse events. Any ideas? No? Yes. So these rats, they didn't get the drug. So we're only teasing them. We're making pretend they will get the drug. So we increase their motivation. These mice got the drug. So this could be, I'm not saying that this is the reason, but it is a possible explanation for the differences between these two models. You can also say that these mice, they don't behave, they don't have any behavior here. It's a different model, but it could be the difference in getting the drug or not. Anyway, so until now, what I showed you is that you see an increase in AMPA to NMDA ratio. So this is a marker of the nucleus accumbens getting addicted to cocaine. This is a long-term effect. When you have a relapse event, Q-induced relapse event, you get a transient increase in the number of channels or in the AMPA to NMDA ratio, but this is a transient increase which goes back to the new baseline, which is the extinction baseline, after more or less 2 hours. And I think we'll continue after the 10. No, I'll talk later about if it's D1 or D2. Okay. כן, תשמע, אתה כאילו אומר שזה לא אותו דבר, לתת לעבר שוק ולהגיד שבן אדם יש לו איזה... זה לא משהו אקוטי, זה לא שכל פעם שהוא מקבל את הסם, זה לא אותו דבר, וגם אני מסכים איתך.
כל שאני אומר שאנחנו אם יראו את הפרעה של הספרים, זה עדיין לא יהיה שום דבר.
כן, אבל אנשים יוכלו להגיד שזה קצת יותר קרוב למצב הזה. זה נכון. תראה, קשה מאוד לגרום לעכבר להיות מודאג מהתוספות...
לא, זה
בעיה, כן. יש ניסיונות, אולי אפשר אחרי זה לדבר על זה, אבל זה מאוד קל לגרום לעכבר מודאג להפסיק להשתמש בקופאין. מאוד מאוד קל.
על ידי כאלה...
אתה רק צריך לתת לה, כאילו, יש מעבדה, לצחוק בעצמה, כן. יש, כאילו, כמו שאתה אומר, העניין, כאילו, בבני אדם, כשהם עוברים את הגמילה או מפסיקים, זה מרצון. הם רוצים להפסיק. אז יש עכשיו מודל של voluntary abstinence. כאילו, התעודות מפסיקות ללחוץ בשביל סם, מרצון. העניין הוא שהמוטיבציה של התעודות להפסיק ללחוץ על הקוקאין היא לא באה מאיזה דאגה או משהו שלילי, אלא להפך מ-reward, נותנים להם אוכל טעים כזה, של תעודות. מבחינת התעודות זה natural reward כזה שהן רגילות לצרוך, אז לצרוך הקוקאין, הן לא יודעות מה קורה. הן מרגישות טוב פתאום, אז הן מקשרות את זה, אבל זה הרבה יותר מבחינתם ישיר לאכול. אז ברגע שאתה נותן להם דרשה של קוקאין, נגיד, אימנת את החולדה, משתמש בקוקאין, קבוע וכאילו דיפנד. אתה לוקח אותה חולדה, במקום סתם כאילו לבטל. אתה משאיר את הדרשה עם הקוקאין, ומוסיף דרשה שנותנת אוכל. תוך יום-יומיים היא מפסיקה ללחוץ על הקוקאין לגמרי. היא תחזור תיקח את הזה של האוכל, כאילו, היא כן תחזור, אבל היא מפסיקה. לא, הניסויים של האלה שלוחצים עד שהם מתים, זה עכברים, שוב, זה כן, זה הגרמה, זה לא כל העכברים. היו עכברים שנתנו להם, נותנים לעצמם גירוי בתוך המוח, בדיוק למדיאל קורבל ששחרר דופמין. אז אלה כן יעדיפו לאכול מאשר לאכול. נקודה אם תיתן לה צ'וייס, היא תלך לאוכל. בגלל זה, עכשיו כאילו מנצלים את זה ואומרים, הנה, נקודה נגמלה מרצון. הפסיקה להשתמש מרצון. שוב, זה לא באמת מה שקורה בבני אדם, אבל זה איזושהי דרך לגרום לחולדה לרצות שזה משהו שהוא לא נחשב בביסטי. עכשיו הוא יכול להיות אפיסטי, לא ברמה של הדופמין, הוא לא חזק כמו סמים, שהם בעצם איזשהו משהו פרמקולוגי שהולך ומשנה את המוח. לא ידוע, יש לו אפקטים פרמקולוגיים משמעותיים. זה מה ש... עכשיו יש כאילו כל מיני ניסיונות, האמת זה מעניין איך המודלים האלה מתפתחים. עכשיו כבר עושים במקום שוק חשמלי, רוצים לבדוק איפה עובר הגבול, כל תעודה, איפה היא תחליט כן ולא. כן, כאילו, אם אתה נותן שוק, אז יש חוקות שימשיכו, כן, ויש חוקות שיפסיקו.
גם זה מאוד מאוד חזק,
שוב, אז מצאתי, אתה לא נותן להם שוק, באמת שתקבוע בהם.
לא, אבל
יש חולדות, כן, חולדות הכי קומפלסים שהיה נבלעו זה להבין לקבל את הקוראים. עכשיו עושים משהו אחר, עושים, אני לא זוכר אם עשו את זה עם אוכל, וגם בהקשר של האוכל והקוקאין. אני חושב שעשו את זה עם אוכל. חולדות מצות מקבלות אוכל ומחכו לאוכל. אבל אם אתה מתחיל לתת דיליי, לוחצות על האוכל, ורק אחרי כמה שניות. באיזשהו דיליי מהאוכל חזרה לאוכל. זה כאילו יש להם איזה decision making של מה rewarding, מה לא rewarding.
אבל טוב, בגלל שלא יחצו אותם
לא, הם יודעים שהדרכה נותנת אוכל. זה לא שהם לא... לא מתאים להם לחכות עכשיו דקה. איך אפשר לבדוק שלא? תראה,
איך עכשיו גם אחר
לא, זה היה ברור. מדברים פה על איך שלא תצליח שעה. אולי יש לי חולדה זה יום ולחר, המשמעות של 10 ו-20 שניות. לא.
האם ריבוד כן,
כן, נלחצו יותר. זה בכלל יש במהלך גם מכורה, או שזה יותר מיידי, כן, זה יהיה יותר קל לגרום לעבודה.
כן, הוא היה הראשון.
תגיד, אתה רואים את זה נגיד, אתה לעומת
כן, ככל שאתה גם יותר מיידי וגם מאפשר לך
יש לו איזושהי טענה שהמאמרים חשובים
וישר, אז הכל ניתן. כן, יש פה בהחלט בגלל הקומפולסיביות, צריך את זה, הקומפולסיביות והאימפולסיביות. אתה במצב פתולוגי, אתה חייב את זה עכשיו. מבחינת המחור, יש הבדל. אם תגיד לו, אני יכול לתת לך את הסם הזה עכשיו, או את הסם הזה שהוא קצת יותר חזק, אבל עוד חצי שעה. הולך לחלל, תביא לי את זה עכשיו.
המאמרים בארץ כל כך גדולים. אני עדיין מופתע מזה שקראתי את המאמר של פיורינג ומה היה בו. המאמר הראשון של פיורינג, שלא הציע מכונת פיורינג. קראתי אותו.
דברים קורים.
אז מאמר הוא...
באמת העניין של המודל זה...
ואז יש לזה 3 עמודים על זה שאתה עושה מפגרים.
זה בעיה.
העניין
הוא שבגלל שזה דימוך מחקר יש אנשים שכל מה שהם עושים בחיים זה לנסות למצוא את המודל. זה המודל הכי טוב. אנחנו, אנשים כמוני שהם פיזיולוגים, מאמינים בפיזיולוגיה של המערכת, אנחנו כאילו לוקחים את המודל שמישהו פיתח. זה מה שיש לנו ביקורת על המודל. אתה יודע, בסופו של דבר, אם השאלה שלך היא מה קורה למוח בהתמכרות, אתה לא מתעסק עכשיו עם הניואנסים של כן קיבל שוק, לא קיבל שוק. אם אתה אומר, איזה שינוי הזה קשור לקומפולסיביות, אז אני רוצה לבדוק שקיבלו את השוק, לעומת כאלה שלא קיבלו את השוק, או קיבלו את השוק וכן השתמשו בשוק. שם אתה יכול...
I propose to consider the question כמשיב, ככה זה מתחיל. זה באמת מבחן טיעון. זה מאמר פילוסופי, אז הוא מדבר על הצרפתים. הוא מדבר על מבחן, ואחר כך הוא סוקר את כל ההתנגדויות שאנשים מעלים לרעיון הזה, הוא מתווכח עם ההתנגדויות. המאמר של המכונת טיעון בשנות ה-30 לא מתמטי לגמרי. אבל בפיתח את המודל של מבחן טיעון הוא עושה את מה שאנחנו עושים היום. בלי כל טיעון. כן, בלי כל טיעון והפורמליזם היפה שכתוב את זה. בעיניי, ואז
תמיד היה משהו
המאמר שם,
חצי כולם ידעו,
הקטע שלו זה להכיר
חצי כולם לדעת.
בעצם בעיית ההגדרה. אני אותה בעיה פתוחה, מהבעיה של גילברט. המאמר הזה מ-50 ורוטר 2
יש כאילו מבחן בסוף, יש מה הקטע מה איך מעריכים אתכם? איך אני אמור להיות?
כן, זה ברור.
איך אתה בסוף?
לא,
ללבן את זה במדע כל כך, אבל...
לצערנו.
זה כמה זמן, ובסוף לקרות.
אבל הפעם אני מתחיל אותו בשבת, כאילו בסופש, ולא משאיר אותו להתחיל ביום שישי. הספקתי בזה קצת.
טוב. יש עוד אנשים בחוץ? אתם כולם קשורים לזה? אני לא... אוקיי. I'll start to go a little bit faster, but if I have quite a lot to show, but I don't have to. I don't have a need to show you everything. So, if you have questions or if something is interesting for you, ask, and I'll give you whatever we have time for. So after we found this phenomenon in the nucleus accumbens, we asked ourselves if this is unique to cocaine. So this a question I get a lot of time from many people, students, other researchers, is what if I found now something that happens in addiction, is the brain now addicted to everything? Is it, if there is a common pathway to all the drugs, then maybe the same thing is supposed to happen if I use cocaine or alcohol or heroin, nicotine. So we wanted to check on two other drugs if we see the same thing that we saw in cocaine. The first drug we used was nicotine, and you can see here that with nicotine we see more or less the same that we see with cocaine. So there is a gradual increase in the AMPA to NMDA ratio. Again, same conditions. So it looks like nicotine is affecting the brain in the same way that cocaine does. When we moved to heroin, the picture was a little bit different. So you can see here, this is the AMPA to NMDA ratio. You can see that one thing I didn't say, that the ratio is the ratio between the peaks. Okay, so you see that the peak gets higher of the AMPA here. When you look here with heroin, you see that the AMPA to NMDA ratio of the peaks is not changed. There is a change in the time course of the NMDA, which is a different story that I will not talk about. But this is something different. So after withdrawal from heroin, you don't see this increase in AMPA to NMDA ratio. If anything, you actually see more NMDA compared to AMPA. This could be related to the fact that cocaine and heroin actually have, even though they're both addictive, they have opposite effects on behavior, on motor behavior. So cocaine is a stimulant. It causes people to move more. Heroin is a sedant. So with rats, you see it very clearly. After a session of self-administration, the cocaine rats will be jumping in the cage, really moving fast, while the heroin rats will sit still and almost not move.
So back to the nicotine, if it's the same effect as cocaine, so if you start with cocaine and then only the installment you give nicotine, will be
Yes. there was a try like 5 years ago to advance a hypothesis that was called the gateway hypothesis, that says that if you get addicted to one substance, it makes you more vulnerable to get addicted to another different substance. It wasn't the experiments were not done so well. So right now it disappeared. So I don't know if this is true. I know it's people think that they make the connection. People make a connection. If you're using one substance, then you'll probably be using another substance. I can give you in the military, I was in the criminal police. I was a criminal police investigator in Messach, and we talked. I mean, I investigated tens of people that were accused with using drugs, and almost all of them, I remember, their main not justification, but the way to convince me that they didn't use the drug is telling me that I don't even smoke. So they make it's not relevant if you smoke or not. If you use the drug, you use the drug. But there is a connection that this connection was not proven at the level of the brain and synapses.
But it's a correlation and effect, I mean, with your humans?
There is a correlation usually. I think the strongest correlation will be between smoking and drinking alcohol. But it doesn't mean that drinking alcohol will make you smoke. It could be related to the social ability. So people that smoke usually smoke together and then they drink together. It doesn't necessarily a causative effect. Yes?
I understand it correctly, but there is a phenomenon that is described about cross-sensitivity.
Yes. I mean, depends on what drugs.
Is dependent on cocaine, but not the other way around.
Yes, but well, when you talk about this, a physiological effect of the drug. If you talk about really getting addicted or behaving like operant behavior, you want cocaine because you were exposed to nicotine before, or the other way around. This was not yet really proven as it should be. Anyway, so in heroin, we don't see these changes in AMPA to NMDA ratio. Yes? No problem, no problem. More or less. They sometimes they press less the lever, mainly because they move less. I mean, during the session they simply, some of them just stop moving. They lay on the lever. Yeah, you open the box and you see the red. But when you check when you calculate the ratio between the active and the inactive lever, so you see it's more or less similar between drugs. Yes. On the brain, I mean, their pharmacological mechanism.
I mean, if you not just smoke, if you give it with an ampulla, if you try to stop.
But when you say similar, you mean do the animals look similar? I mean, if they behave similar? Well, nicotine is not a strong stimulant. No. It's another system. It's the cholinergic system. So no, it's not a strong stimulant as cocaine.
Can you explain maybe the difference between heroin, because this is the synthesis
Yes.
That's why I tried to understand the comparison with nicotine that showed this similar results.
It could be. I mean, it could be. I don't know. I mean, it's just hypothesis, speculating. What was, if we said heroin is not like cocaine, so it's only in the extinction. When we make these animals relapse to heroin, then you see an increase. So this is in vivo. It's not not in slices, but you can see that the EPSP, the amplitude of EPSP is gets bigger after relapse in heroin. So this is similar to what we see in cocaine. Another thing, I talked about metaplasticity. So in cocaine, I talked about the inability to change, the inability of the synapses to change. We see that also with heroin. It's not as strong as here, not as beautiful and clean, but still you can see a weaker ability or lower ability of the synapses to change. So this is a similarity between these two models, these two drugs. But overall, it seems, and there is data I didn't show you, but it seems that the state after withdrawal differs between the drugs, and the state after relapse. This increase in the glutamatergic input into the nucleus accumbens, it seems like it happens in, I don't know if all the drugs, but in many drugs. It seems to be something that is more in common of all the drugs. Now, what about motivational disorders that are not induced by using a drug? So we did two models. One is stress-induced addiction. So I don't know if you know, but people with PTSD, post-traumatic stress disorder, they are much more vulnerable to become addicted than the normal society, the general society. And we wanted to see if we see in rats that went through an acute stress event. So this is one stressful event that we give them 21 days before we start checking their cocaine self-administration. So it's one event. So it's a model of PTSD. We wanted to see if their nucleus accumbens is similar to those to addicted to rats that self-administered cocaine. So we have rats that didn't get the stress, and rats that did get the stress, and you can see that stressed animals press the lever much more frequently. All the blue lines compared to the sham animal. And then when we checked AMPA to NMDA ratio, we saw that the stressed animals were also showed increased AMPA to NMDA ratio. So this is something similar between stressed animals and animals that used cocaine for 2 weeks and then withdraw. Another thing that we are very much interested in the lab, but I will not talk about it a lot, is obesity. So in the modern society, we know there's a very big problem of obesity. Obesity is actually the number 2, how do they call it? The number 2 preventable the number 2 cause of preventable death. Death that could be prevented. Can anyone guess what's the number 1 cause for preventable death? What? No.
Car accident.
What?
Car accident.
No.
Heart.
No. Mistakes of physicians. But so obesity is the number 2 cause. I mean, all the heart problems and diabetes, blood pressure, cholesterol, all that. Anyway, we wanted to see if letting rats develop obesity could cause in the nucleus accumbens things that are similar to what we see in cocaine. So we let rats, just a general, not transgenic or no genetic difference between, I mean, nothing that we know of between the rats. We gave them this high-fat, high-sugar diet for 8 weeks. And at the end of these 8 weeks, we checked the weight gain, how much weight they gained over these 8 weeks, and we saw a pretty wide spread of the weight gain. So we decided to, instead of compare all this group to rats that didn't get high-fat, high-sugar food, we compared the rats that were, we call them obesity-prone, the top third weight gainers, with obesity-resistant, the bottom third weight gainers. Okay, so fat and thin. We compared many things, but I'll show you the AMPA to NMDA ratio. So you can see in the nucleus accumbens that the obesity-prone rats have a higher AMPA to NMDA ratio compared to the obesity-resistant rats, and they also show metaplasticity. So these obesity-prone rats fail to show long-term depression in slices. Actually, the obesity-resistant mice, which seem to have here long-term depression, this LTD is weaker than what we see in rats that didn't get any high-fat, high-sugar food. So it seems like the food itself, independent on if the rat becomes obese or not, the food itself affects the ability of the synapse to change. In these parameters, the AMPA-NMDA ratio and the loss of LTD correlated well with tests that we did to measure the motivation of these rats to obtain the food. So it's another example of an increase or potentiation of the glutamatergic input to the nucleus accumbens in a state of increased motivation. So the next question, now that we see that something is happening in the nucleus accumbens, and you remember there are D1 and D2 medium spiny neurons, we wanted to go into the cell type specific world and see if there is a difference between D1 and D2 medium spiny neurons. I don't have to show you this. Now there has been data for already a long time showing that when you activate with Cre-dependent, you infect D1 medium spiny neurons of the accumbens with channel rhodopsin, and then when you activate them optogenetically, you increase their motivation to obtain drug. While if you do this with D2 So if you put the channel rhodopsin, you activate D2 medium spiny neurons in the nucleus accumbens, you actually decrease the motivation to get the drug. And in these experiments, the light was in the nucleus accumbens. So they infected neurons in the nucleus accumbens and turned the light on in the nucleus accumbens. Two other groups actually showed, so they did self-administration in mice, and then extinction for 30 days, or abstinence. And then they used mice that allows them to record from D1 or D2 medium spiny neurons. And when they recorded this glutamatergic input and measured AMPA to NMDA ratio, they found that actually the synapses that show the difference in AMPA to NMDA ratio are the synapses on D1 medium spiny neurons and not on D2 medium spiny neurons. So it was shown both by the group of Christian Lucher and another group in NIH of Veronica Alvarez. Both showed more or less the same. So it makes sense. You know, the D1 medium spiny neurons are the one driving motivated behavior, so it is much easier for the motivational input from the cortex to activate them because the synapse is stronger, right? So it makes sense. Now, if this is true, what do you think would happen if you now, instead of putting the light in the nucleus accumbens, if you put the light where the terminals are, right? So what would happen if you inject an inhibitory opsin into D1 medium spiny neuron or into the nucleus accumbens, and then put the light in the VTA or substantia nigra? So you take out the direct pathway, right? What would happen to the motivation to obtain drugs? What do you think would happen if you inhibit the direct pathway that is supposed to drive or promote motivated behavior?
You will not have motivation to anything.
Okay. So you would say that you will see a decrease or less lever pressing. But when we did that, and we actually didn't see nothing happened. So if you take this rat, we injected halorhodopsin in the nucleus accumbens and put the light in the substantia nigra in this case, and we put the rats through a reinstatement test. Okay, so this is after they were extincted. We put them into a reinstatement test, let them press the lever, inhibited this direct pathway, and nothing really, it didn't affect behavior.
So we didn't see any motor?
No, we checked.
Say something about the
About what?
I mean, is it 100% sure that it blocks the direct pathway?
No. We can't be sure that it blocked it. But in order to, so the control we did for that, it's not actually a control, but we did the same experiment. Well, we did the experiment without channel rhodopsin, with only opsin, and it looked the same because we didn't have any opsin, right? So the opsin was there. We know from we took slices after we finished. We took slices and verified that the opsin was there, and electrophysiologically, we know that the light should have inhibited the activity of the terminal. We know that. It's not 100%, of course, but when we did it, so this is like the proof that it should have worked. When we did the same experiment, but we put the light in the ventral pallidum, so part of the indirect pathway, then we saw an effect on the behavior. So we were pretty surprised or confused by this, because if you activate the D1 medium spiny neurons that go to the substantia nigra, you generate behavior. But when you inhibit them, you don't inhibit the behavior. Actually, when you inhibit the terminals from the nucleus accumbens to the ventral pallidum, then you're able to inhibit behavior. So something doesn't fit, right? So we started to look into it, and we generated a mouse that expresses red, TD tomato, in D1 medium spiny neurons, and green in D2 medium spiny neurons. And when you look at the ventral pallidum, which is supposed to have only green, you see that there is also red in there. We did a lot of experiments. I'm not going to really talk about it. What time is it? Do I have time? Oh, I still have some time. So we did a lot of experiments, and the bottom line is that the organization of the D1 and D2 medium spiny neurons into direct and indirect pathway, at least for the nucleus accumbens, is not really, there's no really segregation. So when we recorded from ventral pallidum neurons, and specifically using retrograde tracers from ventral pallidum neurons that we know that go to the ventral mesencephalon, and then put channel rhodopsin in D1 medium spiny neurons, we could get in about 50% of the cells of the ventral pallidum, we got input from D1 medium spiny neurons. Okay, almost all of them also got D2 medium spiny neuron input, but this actually means that the ventral pallidum is not connected only to the D2 medium spiny neurons of the nucleus accumbens, but also to the D1 medium spiny neuron. Okay, so the circuitry is more complicated than the classical model, and now it looks like the ventral pallidum becomes a region of integration of information about the motivation to use the drug. And we wanted to see in the ventral pallidum if there is any plasticity occurring in there, in the ventral pallidum, after withdrawal from cocaine self-administration. So just one thing about the ventral pallidum. It receives mainly GABAergic input from several regions in the brain, but mostly from the nucleus accumbens. The two inputs from the nucleus accumbens release GABA. They also express neuropeptides. Interestingly, they release different neuropeptides. The D2 medium spiny neurons release enkephalin and neurotensin, but I will not talk about neurotensin. The D1 medium spiny neurons release dynorphin. They are both, dynorphin and enkephalin are opioid neuropeptides, but they affect different receptors. The enkephalin works mostly on mu and delta opioid receptors. Dynorphin works on kappa opioid receptors. The role of these neuropeptides or of any synapses in the ventral pallidum was not known until recently. It's still not really known. But the first thing we wanted to see if there is anything happening, is any difference between the GABAergic input to the ventral pallidum, if it is changed before and after using cocaine. And it looks like there is a decrease or a weakening of this GABAergic input from the nucleus accumbens to the ventral pallidum. Or actually, there is a general decrease in the GABAergic input because if you check the miniature input to to the cells of the ventral pallidum, you see that the frequency is lower. I don't know if you know what the miniature postsynaptic currents are? Okay. And you know that changes in frequency indicate a presynaptic effect? So if you let's see if I have, yes. So this is what it looks like. And you see that these events have amplitude and they also have a frequency. The frequency of the event depends on the probability to release a vesicle. The amplitude of the events depends mostly on the receptors, on how many receptors you have postsynaptically, even though there can also be a presynaptic component in there, but generally speaking, frequency indicates presynaptic mechanism, changes in amplitude indicate postsynaptic mechanism. So here you can see a change in the frequency, a decrease in the frequency, so it means that there is a decrease in the probability of release of GABA from GABAergic terminals that we don't know where they come from in this case. So they release less GABA. And we also see that as in the nucleus accumbens, here we also see some kind of metaplasticity. We try to induce long-term depression, and we were able to do it in control animals, but not in cocaine animals. Now, what could be the cause for these changes? I told you there are neuropeptides that are released in the ventral pallidum from the nucleus accumbens or other sources. And so we thought that maybe the neuropeptides are there to modulate synaptic release, and maybe these changes in synaptic release of GABA are caused by activation of the opioid receptors. Cut a long story short, we found that when we tried to activate mu-opioid receptors, we were able to inhibit activation of mu-opioid receptors. Successfully inhibited GABA neurotransmission in the ventral pallidum, but not in cocaine animals. So one of the explanations that turned out to be the correct one was that maybe because the synapse is inhibited, maybe there is a tonic inhibition caused by tonic activation of the mu-opioid receptors in this case. And to reveal tonic inhibition, we applied an antagonist of these mu-opioid receptors, washed the antagonist, and you can see that as we increased the concentration of this antagonist, we were able to get a stronger disinhibition of GABA neurotransmission. You can see it here. So this is the control, and the more antagonist we applied, the bigger the GABAergic current became. So, I didn't put here. Okay, so it looks like in the ventral pallidum there is a tonic inhibition on GABAergic synaptic release caused by enkephalin and mu-opioid receptors. Now, are these changes in the ventral pallidum? So what I showed you now is a general effect in the ventral pallidum, and I told you that now we know that there is input from D1 and D2 medium spiny neurons into the ventral pallidum. So we wanted to see if this effect in the ventral pallidum happens on both of these inputs, or maybe only on one of them, and then this could mean that there is a differentiation between the actions of these two inputs in the ventral pallidum. So we repeated the same protocol, only now instead of electrically activating the general input to the ventral pallidum, now we used optogenetics. And you can see that in saline animals, both D1 and D2 medium spiny neuron input to the ventral pallidum can undergo LTD. You can see green is, I think, D1. No, yes, green is D1, red is D2. Both of them undergo LTD, but after they undergo extinction, you see that the D2 can no longer show LTD. The D1 are still normal, the D1 input to the ventral pallidum. Now when we, again, try to see if this is a mu-opioid receptor effect, so we used this blocker of mu-opioid receptor, CTOP. First of all, when we used cocaine animals, control animals, sorry. When we used control animals, CTOP prevented the generation of long-term depression. Which makes sense. You can't activate the mu-opioid receptors. And when you take cocaine animals and you apply CTOP, you see that the input from D2 medium spiny neurons is disinhibited. The D1 input is actually, well, nothing really happens here, but if anything, there's a trend in the opposite direction. But we don't know why this happened. But this result shows that the mu-opioid receptor, this inhibition, tonic inhibition on the GABAergic input from the accumbens to the ventral pallidum is mainly on D2 medium spiny neuron input to the ventral pallidum. So, in a sense, you can say that the ventral pallidum in normal animals, not addicted, gets two inputs from the nucleus accumbens, D1 medium spiny neurons, D2 medium spiny neurons. The D2 medium spiny neurons contacts many more cells, and therefore it can be perceived as a stronger GABAergic input. And now, after cocaine, after extinction of cocaine self-administration, this D2 medium spiny neuron is selectively inhibited. So it becomes weaker, and if you look at it from the perspective of the D1 medium spiny neurons, the relative influence of the D1 medium spiny neurons on the activity of the ventral pallidum becomes bigger, right? Because the D2 MSNs are inhibited. Now, is there any behavioral relevance? I mean, so what if this happens? So, as I told you already, when you activate, yes?
So, in general, can you go, yeah. So, like if we take Gary-Bergman type of approach, in this case, it just demonstrates that the pallidum just gets less inhibition. In both cases, you just get GABA.
Yes.
Unless we know something about the target or the relative kind of encoding of these two pathways, why do we really care? So if we just increase GABA,
if you generally increase GABA in the ventral pallidum,
would that just kind of solve the problem? Is it just not enough GABA, or is it something more specific?
I think it's more specific. Well, if you inhibit the ventral, if you inject GABA agonist into the ventral pallidum, you take out motivated behavior in general. When you activate D2 medium spiny neurons, optogenetically, as I showed, it inhibits motivated behavior. So you increase GABA.
You don't project only to the D2,
The D2 medium spiny neurons, it projects almost only to the ventral pallidum. But when you activate D1 medium spiny neurons and they increase activity or motivated behavior, when you, well, now I will show you that the D1, there's a difference between the D1 and D2 input into the ventral pallidum. Even though they both release GABA, their effect on behavior is different. Now I will show it. So it could be, I mean, if you just increase GABA generally, I think anywhere in the brain.
No, I'm thinking so you could do the experiment of, let's say D1
Yeah.
and OD2, the channel blocker,
Yeah.
and then stimulating the D2.
Yes.
So according to this picture, in the cocaine animal, it doesn't matter if you only activate the terminals in the D2.
Mm-hmm.
It shouldn't matter if you activate D1 or D2. If this is the same,
Unless they have different targets. Of course. Yes. We will try to understand that in the future. Yeah, but there is some level, should be some level of common target, because like 90% of ventral pallidum cells get D2 input, and 50%, so the numbers. But I don't know. We don't know if there's anything special about this. We know nothing about that. Okay. So what's the behavioral relevance? So Mickey suggested the ideal experiment, but we didn't do the ideal experiment yet. We did something similar, not perfect in my opinion, but so what we wanted to see if activation of the D1 medium spiny neuron input to the ventral pallidum is the one that generates the promotes motivated behavior, because we know it's not the input to the substantia nigra. We inhibited that input and we didn't see anything. So we use the DREADS system now. So you know what DREADS are? We use the excitatory or stimulatory DREADS in the nucleus, in the D1 medium spiny neurons, and well, first we injected CNO, IP. Okay, I'll finish with that. We injected CNO IP, and mice had stimulatory DREADS in D1 MSN. Not surprisingly, it has been shown before, you see an increase in motivated behavior. What we did next was to take another group of mice and inject them with the stimulatory DREADS in D1 MSN and inhibitory DREADS in the ventral pallidum. So now when we inject CNO, we simultaneously activate D1 MSNs and inhibit or shut down the ventral pallidum. And in this group, we didn't see the effect of the D1 medium spiny neurons on lever pressing. So it seems like this effect of D1 medium spiny neurons requires an active ventral pallidum. Okay, so the terminals of D1 medium spiny neurons in the ventral pallidum, maybe these are the ones that promote drug seeking. We did the opposite. We did two MSNs. This time we used the inhibitory DREADS in D2 MSN. So when we inhibit D2 medium spiny neurons, again, not surprisingly, we increase drug-seeking behavior. But then, again, when we shut down the ventral pallidum, we lose this effect of D2 medium spiny neurons. So the effect of D2 medium spiny neuron, not surprisingly, is mediated through the ventral pallidum. So what we're thinking right now is that the D1 medium spiny neurons input to the ventral pallidum is a go signal, because when you activate it, you generate motivated behavior, and the D2 medium spiny neurons is a stop signal. And in normal animals, this stop signal is strong enough to prevent unwanted motivated behavior. But in cocaine, there's a specific inhibition of the input from these two medium spiny neurons into the ventral pallidum, which may mean that the stop signal is not strong enough, and then the go signal can really make the ventral pallidum somehow, we don't know yet how, but somehow drive motivated behavior. So this means that the problem in addiction is not an increased desire to go for the drug, but a decreased ability to modulate the behavior, if this is correct. And I think I'll stop here. I have more on dynorphin, but we'll stop here. Questions? Comments? No? Okay. Yes.
So, in the previous hour, you made a statement about metaplasticity.
Yes.
And right after that, you've shown a figure that shows that you increase in plasticity after the
Increase in the AMPA to NMDA ratio.
following
Yes.
relapse, right?
Mm-hmm.
This sounds strange.
Yes. I've been asking myself and everyone I know in the field, are we really seeing metaplasticity, or is it an artifact of what we do? I mean, it is true that after cocaine, the protocol that we use to generate LTD doesn't work as well. I don't know if this is what if something else happens in the brain, then maybe what we see is irrelevant, because maybe the synapse can change. The main thinking behind these experiments is that if you want to treat people by intervention, then your only way to depress synapses is by, right now, is to give them a protocol that that depresses the synapse, and this doesn't work. The protocols that we use, they don't work. So the synapse, it does lose some of its ability to change, but there are definitely several ways to generate plasticity. So maybe other pathways or other mechanisms are not affected by cocaine. It's possible. Okay. למה כאילו נכנסו למערכת הזאת בתור reward system? כאילו, למה התלמוס לא חלק מהreward system? הם מחוברים לתלמוס, איפה נגמר הגבול? איפה כאילו? כן, עכשיו היא לא נגמרת. הכל ביחד עובד. לא, זה לופ, כן, אבל זה לופ שהוא הוא מאוד, אתה יודע, אולד סקול כזה. כל פעם שיוצא איזה מאמר, אתה יודע, נגיד סבתים. אני מקבל את הג'ננה, כאילו, הם מצאים כאילו, אנשים מוצאים שני תאים שהולכים מהפריאנום חזרה לקורטקס, חופרים, מוצאים בסוף משהו שהשתנה הדוגמה. הוא כאילו מפרסם מזה, הוא אומר, שני תאים, יש לך 100 תאים שעושים הפוך. כן.
משהו כאילו מאוד, לא יודע, זה נראה כאילו אין שם שום
שאין שם? איפה? מה זה שם?
זה כאילו מדהים אותי שאתה עושה ניסוי שבו לא משנה איזה תא אתה לוקח, אתה רואה שינוי
איזה סוג תא פה?
לא, כאילו יש D1, D2.
נכון.
לא משנה, זה לא שאתה צריך ללכת ויש אלה עושים פרודקשן לאלה ואלה עושים פרודקשן לאלה ויש כל מיני, זה כאילו כל הסינפסות עוברות מודולציה, זה כאילו כל האוכלוסיות האלה לגמרי הומוגניות, מקבלות אינפוט מכל מקום, לא אכפת להם שום, זה פשוט אותו תא משוכפל מיליון פעמים, זה מה שזה.
היה מאמר שכאילו ניסה להוכיח בדיוק את ההפך למה שאתה אומר, מאמר של בונצ'י, ב-2012, פרסם בנור, כאילו סרט, glutamatergic input, different glutamatergic input affect addiction differently, והם מראים שם, הם כאילו הזריקו צ'אנל רודוקסין במיגדלה, בוונטרל היפוקמפוס ובמיגדל פרפרונטל קורטקס. לא שגם הוא עשה את זה אחרי. כן, הוא עשה את זה כמו שלו יודע לעשות. D1, D2, וכאילו כל מה שאפשר לעשות הוא עשה. הם בעבודה הראשונה, ב-2012, הם לא עשו D1, D2. זה היה בחוזות, הם רק רצו את האינפוטים, כאילו להראות שכל אינפוט עושה משהו אחר. והם מראים, כאילו מה הם עשו? הם מדדו אמפה NMDA ratio של כל אינפוט בנפרד, והקומבנט בלי D1, D2, וראו שאחרי המודל, מזריקו, לא חשוב, אז הסינפסות של הפרפרונטל קורטקס השתנו ככה, אלה לא השתנו, אז זה כן השתנו, ואז הם אמרו כל אחד אחר, ואז הלכו ועשו in vivo optogenetics, כי הם נטפלו שם מאיזושהי סיבה לוונטרל היפוקמפוס, פשוט בגלל הוא נתן להם, הם עשו של ושם הוא מגיע, לא חשוב, זה מין הטיה, אבל והראו שהוונטרל היפוקמפוס שאתה נותן לו אינביציה, פחות זה מתנהג. עכשיו, הפיגר האחרון של המאמר, שהוא לדעתי בא מרביואר, שביקש ממנו לעשות, מראה שזה לא משנה מה אתה עושה, אתה מעלה את הגלוטמט ויש לה הכל אותו אפקט. כאילו, למרות שאתה רואה שינויים באמפה NMDA, בסוף, כשאתה שם צ'אנל רודוקסין ונותן תדר, לא משנה מאיפה מגיע הגלוטמט הזה. אתה עושה שפריץ, זהו. אז כאילו, כל המאמר הזה שהמטרה שלו הייתה להראות שונות, בעצם בסוף מראה, אין כלום.
זה מה שאני
זה תכנית מצדיק רעש, כאילו אם תכנית רעש שהוא מצדיק חלק,
כאילו למי שבא אולי מהקורטקס, למי שבא מהקורטקס, שם כל חייל הוא ככה, והוא שונה לגמרי מהחייל שלידו, כולם מסודרים בעמודות גם וזה, אתה נכנס למטה וזה הכל כזה מרק שאתה...
אבל זה מרק בגלל שזה מרק, אבל זה מרק בגלל שאנחנו לא יודעים להסתכל עליו.
לדעתי, ההרגשה שלי, תראה, הסטריאטום הוא לא...
למה צריך כל כך הרבה תאים?
למה צריך כל כך הרבה
כולם
תאים?
עושים אותו דבר.
תאים לא צריכים לעשות אותו דבר, כדי שאם תכניס רעש שהוא מספיק חזק, אתה תראה אותו בכולם. והם יכולים שכל אחד יתנהג קצת אחרת, ויהיה לו דקויות מאוד ברורות, בסוף אם תעשה רעש שהוא ממש עוצמתי, אם תיתן לחיה להזריק לעצמה כמה קוקאין שהיא רוצה. או שאתה תראה את זה כזה, איך שתחפש את זה. למרות שדווקא פה הראית שלא רואים את זה, שאתה מחפש את זה, לא רואים את זה בכל מקום שתחפש את זה, אבל כאילו, אם זה הסדרה שהוא ממש עוצמתי,
כאילו זה כאילו אומר, כל מה שאנחנו יודעים לעשות זה go ו-no go. אבל זה לא מה שאנחנו עושים בחיים, נכון?
כן, בדיוק.
זה לא אומר.
אנחנו יודעים לעשות go ו-no go עם
שחולדות יודעות
תמיד.
לעשות go ו-no go.
מה?
זה מה שחולדות יודעות לעשות no go. דברים.
אבל גם חולדות זוכרים, יש להם go על זה ו-go על זה ו-go על זה
יש להם
ו-go
שם
על זה.
מתנהגויות
זה
ממש
go.
פשוטות.
אז זה לא מסתדר.
גם רוב מה שהם עושים הוא לא, כאילו, לא נכנס יפה לתוך פיזיולוגיה, כאילו, רוב מה שהם עושים נכנס לפעילויות מאוד מורכבות.
כן.
לא, הם עושים דברים שהם נורא דווקא להפך שהם כמו הרגלים, אתה יודע, עכבר מה הוא עושה, אתה מסתכל עליו, הוא הולך חופר, הולך מריח, הולך חופר, זה בערך מה שהוא עושה, הולך לאכול, הולך לשתות, חופר מריח. זה כן משתדל, אבל יש הרבה כאלה דברים.
כן. לא, אבל זה... תשמע, יש פה... יש את התיאוריה של הספיירל, אני לא יודע אם אתה מכיר את זה, שכאילו כל דבר, כל הביט שאתה מייצר, שכאילו נגיד אדיקשן, כשהוא נכנס לזה, הוא מתחיל בכי ונטרלי, ששם יש הרבה דופמין, הכי הרבה דופמין נגיד, שזה גם הכי...
זה back propagation כזה?
לא, אז כאילו יש לופים עם הוונטרל מזנצפלון הזה, כאילו מהקומבס של שהוא הכי למטה, זה הולך ל-VTA, ואז משם, כאילו, אני חושב שזה בדרך כלל, לא יודע, לא הגדירו, אני חושב, בצורה נוירוביולוגית, זה מודל פסיכולוגי יחסי, של ג'ורג' קוק. כאילו, בסוף המידע חוזר לקומה מעל השל, לקור. עוד לופ הולך לסטריאטו, בסוף מגיע לדורסל סטריאטו, ושם זה מתקבע בתור הביט. יש כאילו מין ספיילר כזה. כאילו, זה לא, יש שם איזשהו סדר. אבל, אתה יודע, ברגע שאתה מתחיל לחקור דברים כאלה שהם, אתה אומר, אתה צריך פה גשטל. יש לך משהו גדול שקורה, אתה מתחיל לחקור את זה עם כלים עזוב אותה מרוב עצים אתה לא רואה את היער, מרוב עלים. אתה כבר כאילו בעלים ובזה, אתה צריך להבין איך היער הזה גדל. אז אני חושב שזה הדרך כאילו, זה הדרך הנכונה.
אני אקצר לכם ממש את זה. אני יכול, שאלה עליך לגבי הריוויו שאנחנו הולכים להגיש תחילת שבוע הבא. האם אתה, זאת אומרת, בהנחיות, אמרת, אתם יכולים לעשות את זה בצורה כזו של כרונולוגית, להתמקד קצת בספרד, להתמקד קצת בשפה אחרת, התחלנו לכתוב, יש לנו, אנחנו פרוסים על תקופה מאוד ארוכה, כלומר, מהגילוי ועד היום, אבל לא, כאילו, יש כרונולוגיה, יש דברים שגילו קודם.
זה לא כל כך אכפת לי.
לא, העיקר שיהיה...
אני רוצה שיש שתי נקודות מאוד חשובות בתוך הדבר הזה.
אוקיי, אבל לא סביבן לעשות את כל הניסוחיאדה. סבבה, מעולה.
יש לכם יד חופשית באופן עקרוני, זה לא כל כך...
תודה רבה.
זה? איזה קורס? זה? כן.
יש לנו 3 אסיינים, ואחד מהם נורמודולציה.
חבל, לא הגעתי בדיוק נורמודולציה כאילו בקטע של סרוטונין ודופמין או נורופפטייד?
לא, אז אני